Let me just start by welcoming everyone and introducing myself. For those of you who don't know me, my name is Lisa McCoy, and I'm the CEO for Defeat Duchenne Canada. And we're really pleased to have our friends and colleagues from Solid Biosciences with us tonight to share some updates with you. This is certainly one of the things that we really enjoy doing, is being able to share information with our community. So just before I pass it over, I'll just tell you a little bit about Defeat Duchenne Canada. Our mission is, well, first of all, we are the country's only national charity dedicated to ending Duchenne muscular dystrophy. And our goal is to provide leadership in research, advocacy, and support to ensure that every Canadian affected by Duchenne can live a long and active life. And our long-term vision is a future without Duchenne, which I'm sure many of you share. Last year, we launched a new strategic plan, and we focus on 4 strategic pillars, 4 areas of work where we feel we can make the greatest impact for the community. And they are research. So we fund world-class research that has led to many new discoveries over our 30-year history. Advocacy, so that we can be there beside families affected by Duchenne and advocating with them for all the many needs they have. Education and support. So we really want to make sure that people who are in need of information about Duchenne have a reliable place they can go to find that information, and Defeat Duchenne Canada is that place. And also community engagement. We engage with the community in many different ways, whether it's through our fundraising activities, our education activities, our support groups. We love to hear from our families. Hearing from our families really helps to guide our strategy and ensure that we're always focused in the areas where we can be of greatest help to you. So, Solid Biosciences, please welcome our colleagues from Solid Biosciences. We do hope that we will have some time for questions at the end of the presentation, but I would encourage you to please use the question and answer function within the webinar to record any questions you might have. And then we will make every effort to address them at the end of the webinar. If for any reason we can't get to all the questions, we will certainly capture them and send them to our friends at Solid Biosciences, who I'm sure will be very happy to send us some responses that we can share with you. So without further ado, let me start by introducing Annie Ganot, Vice President, Co-founder, and Head of Advocacy for Solid Biosciences. And Annie, I will turn it over to you to introduce other members of your team.
That's great, Lisa. Thank you so much. Thank you to the entire team at Defeat Duchenne Canada, and of course to the community for joining tonight, or if not tonight, on the recorded posting that will go to Defeat Duchenne Canada's website following this webinar. So I just want to do a special shout out to the patient community and everybody that participates in clinical research. We know that this is a huge challenge, and we're going to do our best to try to take everybody through our update today. Next slide. So I am joined by my colleagues, Pat Gonzalez, who is a Senior Director in Clinical Science, and also Matt Harmelink, who is the Medical Director and the Therapeutic Area Head for Neuromuscular. Next slide. As a publicly traded company, we may be making some forward-looking statements. Next slide. That's great. Thank you so much for that. And I shall turn it over to my colleague, Matt Harmelink.
Yeah, so I get to start us off with discussing Solid Biosciences in general. We as a company are looking at pioneering the next generation of precision genetic medicines with multiple clinical stage assets. Obviously, tonight we're going to be talking about a Duchenne muscular dystrophy program, but in the neuromuscular realm, we also have Friedreich's ataxia, And we also have an expansion then also into cardiac diseases, as well as even the idea of having delivery, looking at capsid libraries in other areas, focusing on, for example, manufacturing purity, immunomodulation, and other areas. Next slide. In this clinical stage, as we talk about tonight, we'll be talking about our SG003 program for Duchenne's, where we're talking about this first in human data that was just released. But as you can see here from the pipeline, we are also looking at a FA program, Friedreich's ataxia, with first patient dosed in the second half of this year, and then also our cardiac program coming into the clinic hopefully very shortly as we go forward, not to ignore the idea of the capsid library, which makes this all possible of having capsids for gene therapy as we move that program forward as well. Next slide. Now, as we all know, Duchenne's is a disease of impaired muscle integrity and dysfunction. And when we talk about muscle integrity, it's the ability of muscle tissues to remain structurally and functionally whole. And the loss of this muscle integrity we can see by having factors that are normally inside the muscle cells leak into the blood. We can see this as we look at actual muscle fibers themselves. The membranes can lose essential proteins and break down over time. And we can also see that early signals of muscle integrity decline have a negative predictive value for certain organs such as the heart. And so that's why treatments that impact on muscle integrity are key for patients with Duchenne's. Next slide. And with that, I will turn it over to Pat Gonzalez to talk about our study.
Thanks, Matt. And thank you to the entire community for tuning in tonight or whenever you may be watching. We're so grateful to be able to have this opportunity to be able to dive into these data, which have been very encouraging that we just released within the last couple of weeks. So what we'll do tonight is to be able to go through really what is an overview of SGT-003, a bit of background on this therapy itself that we are investigating for the treatment of Duchenne muscular dystrophy, going into the clinical trial that we are currently running, and then the data that we have recently announced and generated, which are the first results from our INSPIRE Duchenne study of SGT-003. So starting with a background on SGT-003 itself, taking it back even a step further is thinking through what are the pieces of an AAV microdystrophin gene therapy that are critical that really drive how it's going to perform once we are looking at this as a therapy. And so AAV is an adeno-associated virus. And so this is a way that we're able to use to be able to deliver a therapeutic transgene to a body to be able to replace something that's missing. And so when we think of each of these different pieces, what do they really mean? The first is the capsid itself. So this is the AAV, the type of AAV, and this is how we're going to actually be delivering. Then we need to have a promoter. The promoter is going to drive the expression, meaning that we're going to have expression of what we're trying to deliver in specific tissues. And so for our indication, when we're looking at Duchenne, we're going to be looking at driving expression in skeletal muscle, cardiac muscle, lower limb, upper limb, really muscles throughout the entire body. And then most importantly is going to be the transgene. And so this is what you're actually delivering. So when we think about a treatment for Duchenne, we want to be able to get at the root cause of the disease, which is the loss of a functional dystrophin protein. With an AAV, what we're doing is actually delivering a form of dystrophin that can fit within this AAV to make up for that lost dystrophin. So specific to SGT-003 are each of these different pieces that we're using for this approach. And we consider this a next generation gene therapy approach really because of these unique components. I mentioned how critically important the microdystrophin actually is, this transgene that we're delivering. And so because the capacity of an AAV is much smaller than a full-length dystrophin protein, we need to make important choices on which pieces can be included in a transgene that's being delivered. And so as we know, the full-length dystrophin has many different components that are going to be of critical importance for its function. Some of these are going to be those that are important for structural function, which is really key for a dystrophin. And that's the intracellular side here that can actually bind the actin binding domain, the extracellular side, which links up to the membrane through this dystroglycan binding domain. And when you have a version that has these two components and these internal pieces, you're able to have that structural stability of a dystrophin being replaced. However, all of these internal pieces can also serve different functions. And the key piece that's included in our SGT-003 microdystrophin is what's called this nNOS binding domain. And nNOS stands for neuronal nitric oxide synthase. It's an enzyme that's actually responsible for resulting in increased blood flow to the muscle when it's located in the right place and activated when necessary. And so we'll go through a little bit more on that in a minute, but it is a unique component of dystrophin in that this ability to localize to the membrane, to actually be located where it's necessary, is dependent on this domain that we are including in our microdystrophin. And on top of that microdystrophin, we go back a step to that delivery vehicle. And so different versions of AAVs have a different ability to get to the different types of tissues throughout a body. Because we're looking at this for Duchenne, we need to be able to administer this systemically so that we can get to all muscles, not just certain muscles over others. And so as we developed this SGT-003 product, we wanted to see, is there a way for us to use a version of an AAV that can get to muscle even better than others? And so what we did was had a rationally designed muscle tropic capsid, so meaning that a version that can get to muscle better. And that's what we've used here for our program, because what we were able to see is that in this version, we can get multiple fold higher levels of these particles to muscles throughout the body. So with that, we consider this our next generation approach, and we'll go into some of the details about what we've characterized preclinically first and now in our clinical trial. I mentioned how important nNOS is, and these are some of the first pieces that we want to highlight before we look at the data that we've generated. And it's really because we want to call out what the different things are that when this protein is present in the right location, it can be responsible for. So when dystrophin recruits and stabilizes nNOS at the membrane, it is very critically important for the ability to actually improve the blood flow by actually resulting in what's called nitric oxide. And so when nitric oxide is produced, you can have vasodilation, which means blood gets better to where it needs to go. With that, you're able to supply your muscles with energy, and you can prevent things that happen when you don't have this process. So if you weren't able to get the energy that's needed and the oxygen that's needed, you undergo what's called ischemia, which is literally the lack of oxygen. And in that, you can have injury to your muscle, which then can result in damage over time. With the restoration of nNOS to the membrane through an approach like this with a microdystrophin, the hope is to be able to address this specific aspect and actually protect muscle even further. And so going through just a couple of pieces of non-clinical data that we've generated prior to the clinical trial, what we wanted to do was really characterize both this capsid and this overall construct that we put together before going into humans. And so one study that we did here is looking at a comparison between a first generation naturally occurring capsid called AAV9 versus this next generation capsid AAV SLB101 in a monkey study. And so what was done was that separate animals were administered equivalent doses of each of these capsids. And what we can see is that we have multiple fold higher levels that are getting to the muscle in the AAV SLB101 compared to AAV9. And so this was really showing that where we want to target, we're getting more of the capsid. At the same time, we also wanted to see, can we be able to actually reduce what it does not need to go to places? And so one of the tissues that when you administer something systemically, you get a lot of particles going to is the liver. And so what we can see is that with this capsid SLB101, we actually have about half of the amount getting to the liver compared to a first generation capsid like AAV9. So these combination of results, getting more where we need to have it and less where we don't, really gave us an initial confidence in being able to want to move this program forward. In addition, one of the key comparisons for us is going to be looking at a full construct comparison, and again, compared to a first generation capsid. And so what we did was ran a study again in monkeys where we assembled an AAV RH74. So this is another first generation capsid with a CK8 microdystrophin. So actually with the microdystrophin construct that we were including in our SGT-003, and we compared that against an AAV SLB101 with CK8 microdystrophin, so essentially SGT003. Similar to the results that we saw from the comparison of AAV9, here we also have 4 to 6 times higher levels of microdystrophin expression that we're seeing in these skeletal muscle and cardiac muscle tissues. And so together, these results really gave us that encouragement and overall rationale to be able to move this program forward quickly. As I mentioned, our first in human study that we're currently running, evaluating SGT003 in the clinic, is called INSPIRE Duchenne. And so I'll go through a bit of an overview of what that study is and these first set of data that we've generated, especially in muscle biopsies. And then afterwards, I'll hand it over to my colleague Matt again to be able to go through more of the clinical data that we've also generated here. As a quick overview of the study, this is now being conducted in the United States and Canada, and we have also been granted regulatory approval to move into the United Kingdom and also Italy. And so we're very happy to be able to discuss this study specifically with you today, as we do have this study active in Canada at a clinical trial site, which is Sick Kids up in Toronto, and we'll have a contact information for the site at the end of this presentation as well. So this study is a single dose study where we are administering SGT003 at not wanting to evaluate different doses, but at the dose level that we believed could have been optimal for that single dose effect based on our non-clinical studies. Currently, with an AAV-based gene therapy, we consider these one-time treatments because we generate antibody responses after that initial administration. And so with that, we wanted to have confidence in the ability to have an effect that could be potentially beneficial from that single dose that we're evaluating, which is why we chose the dose that we moved forward with. The study is an open label study, which means that everyone is getting treatment who is enrolled in the study. And currently we have 2 cohorts that are enrolling between ages 4 to 11. And so both cohorts are actively enrolling in the study right now. Importantly, because this is an AAV-based gene therapy, we want to look to make sure that we maintain safety primarily as we're moving forward with such an approach. And for that, what we have what we've chosen for this is increasing steroids as the immunomodulation in this study, but only increase steroids, whereas some programs use a combination of multiple different factors. And because this is our first in human evaluation, what we really want to do, as I mentioned, is focus on safety. We'll be using that as a primary objective here to make sure that we're very conservatively and comprehensively monitoring safety in the study, and then also looking to see different factors that are secondary objectives here for that potential efficacy of the product, things like the expression of microdystrophin, which we'll be talking about here. And then later in the study, we'll also be looking at things like functional changes using ambulatory functional endpoints like time test, North Star ambulatory assessment, stride velocity, and others. And quickly, I'll just go through the eligibility criteria that we consider key for the study. I mentioned these 2 cohorts of ages 4 through 11 divided between the 2. What's important here to also emphasize are those who are going to be included in the study. Unfortunately, because of an identified risk of having an immune response against a piece of a protein that you may have never seen before, we do have exclusion criteria for specific genetic variants in the DMD gene. And these are deletion variants specifically in exons 1 to 11 or 42 to 45. So this means that if someone were to have a deletion mutation that starts at, say, exon 5 and goes further than 11, they would be excluded from the study. So any piece of these exons that are in a mutation that's a deletion mutation, those patients unfortunately would be excluded. We're hopeful that we can learn from the field and evaluate these sorts of criteria further, but because we want to start very conservatively and maintain safety and evaluate that comprehensively, these are the criteria that we have begun the study with. We'll also be looking at functional criteria specifically in this older cohort because we want to make sure that we're able to look at these ambulatory functional assessments as we move forward with the study. And then from there, because this is an AAV gene therapy, as I mentioned before, antibodies against the AAV could actually prevent the efficacy of the drug. So it could prevent it from doing what it needs to do. And so one of the important criteria as well is being negative for AAV antibodies. And so this is looked at in that screening period. We also want to make sure that we are inclusive of patients, even if they have looked at many other different therapies in the past, investigational or commercial. What we cannot accept are those who have had a gene therapy in the past, because again, this would result in antibodies and a response that you may not be able to get a benefit or could pose safety risks in the study. However, if anyone has been on an exon skipping therapy, vamorolone or givinostat, they would need to go through a washout of 12 weeks, so not being on that for 12 weeks, but then would be able to screen for the study. So they would not be excluded, they would just have to go through this washout. And then the last key piece that we require for the study is for patients to be on a stable daily dose of oral steroids for at least 12 weeks. And this is, again, just to maintain a bit of a lower variability in the study as those who come in, so we can monitor potential changes over time from there. And now getting into an overview of what we have just announced recently have been an update on the first participants who have been enrolled in the study and have passed these important milestones. And so this is an overview of those who we have announced. We had 6 patients dosed here who are highlighted. Actually, at the point of this announcement, we had a 7th patient who was also dosed, so we were happy to announce that as well. Within these total number of patients, we have 6 who have passed the 3-month time point. And because of that, they're going to be the ones that we're highlighting here today from this initial evaluation of our biopsy result and biomarker results as well, because that's that first real milestone in the study. And then we'll be providing a comprehensive safety evaluation of these patients as well. And so we're very encouraged and very excited to present this slide here, which is really an overview of what we saw from these muscle biopsy results from these initial 3 participants dosed in the study. I'll go through each of these pieces to make sure that they're clear, but really what we're separating from the top to the bottom are the total amount of microdystrophin that we are seeing produced in these muscles on the top using multiple methods, and then on the bottom, how that microdystrophin is distributed throughout the muscle. And so when we're thinking about the amount of microdystrophin that we're making, we want to be able to compare that to a relevant standard that gives us some context for what that number is. And so what we do are perform these methods, this one called Western blot on the left side, where what we're doing is looking at that microdystrophin protein and comparing it to a pool of what are considered normal non-dystrophic muscle that are dystrophin positive. So between multiple people and all of us who are on the panel today for this webinar, if we were to look at each of our individual muscles, we would see that we all have different levels of absolute dystrophin. So when we take a pool of muscles, we can then get what can be a more accurate reference as to what that average might be. And so that's what we're comparing against. And so when we look at these values, what we saw were a mean of 110% of normal dystrophin. And so what that means is that in that pool, we would normally have 100% of normal dystrophin. And so this is actually just above that. And so a very encouraging result to show that we're essentially getting back to these levels, which could be comparable to what would be normally in a muscle. This ranged from 84% of normal up to 135% of normal between these 3 participants. So we actually had quite an even range across the 3 around this mean. And so again, emphasizing that if each of us have a little bit different of an absolute dystrophin level, this might be what you would even kind of expect from that. When we used an additional method, what's called mass spectrometry, we were also able to see quite similar results with a mean of 108% of normal dystrophin, doing that same comparison back to a normal pool, with a range also 53 to 152% here, right around that mean. And then importantly, when we look at this from that distribution of microdystrophin, we think of this as a really important factor because it's very important to be able to make enough of the protein, but you need to be able to see it throughout the muscle. If the protein's only being made in these small focal areas, you're not going to be as protective as if you can actually be widespread and have that distributed evenly throughout. And so we were very encouraged to see these results, especially because what we saw were a mean of 78% of positive fibers for microdystrophin, and that range was 70 to 88%, so still quite evenly around the mean. And if we look at a representative image here of what this assessment is, it's that in all of these muscle fibers, which are these circles throughout this entire image, we can see that that vast majority are positive for expression of microdystrophin, which is in red, at the membrane. So it's telling us two things. We're seeing that the protein is actually being expressed throughout the muscle, and that the microdystrophin is functioning on this molecular way, the way it's supposed to, because it's localized to the membrane, and that's where it's supposed to be binding. So if we also go a step further from there, what we want to start doing are looking at other biomarkers that could give us an idea of something that this microdystrophin is doing. And so the first part that I mentioned already is showing that the microdystrophin is where it needs to be in the muscle fiber and that it's widely distributed. But if we start looking at additional proteins, one here called beta-sarcoglycan, and then here the nNOS that we mentioned before, what we can also see is that we have corresponding increases in each of these proteins that go along with the microdystrophin expression that we're seeing. So here for beta-sarcoglycan, this is a protein that's associated with dystrophin and part of this dystrophin-associated protein complex. When dystrophin's absent, it's mislocalized and downregulated from the membrane. And so what you can see here in these two images are that you have this low level here at the baseline, but post-treatment, you have an increase in high intensity of this protein that really corresponds well with your microdystrophin. And so these would be the same fibers here where you have microdystrophin, you have this increase in beta-sarcoglycan. Similarly, what we saw from this nNOS activity stain, so this is using a different method where we're looking at activity from an enzymatic level. What we can see in the baseline is that nNOS really is not present at the muscle membrane. It's essentially zero, and that's what the baseline quantification showed us here in the very, very low levels. But we have a substantial restoration of nNOS that again corresponds well with our microdystrophin expression. And you can see those circles right around the membrane being positive for nNOS activity staining, just like our microdystrophin and the beta-sarcoglycan. So encouraging results on that molecular functionality level of the protein doing what it's supposed to be doing. And this really all comes together when we look at our muscle in one different way, and it's what's called vector genome copy analysis. And so when we're administering SGT-003, the dose level that we're giving, we actually are able to quantify the amount of the genome copies that we're administering. And so with this being the dose, we can then look back at the muscle and see in each of these per nucleus quantifications. So a muscle fiber can have multi-nucleated, they can be single nucleated, they'll have a bit of a variability here. What we want to see is, are we getting at least one copy, and how many more than that are we getting to each of these nuclei to be able to get an understanding of what our expected expression would be. And so we were very encouraged to see that we had high numbers of copies per nucleus, even across all three of these participants, with a mean here of 18.7 copies, so quite robust levels. That then corresponded with microdystrophin expression, so really putting the story together of each of these two pieces corresponding with one another. And so from that, I'm going to hand it over to my colleague, Matt, to go through some of the more biomarker results that we've analyzed from these patients as well.
Thank you. So as I sort of mentioned in the beginning, there's these different markers that we can use. And as we talk about biomarkers, I'm going to start off with looking at what are biomarkers in general. I think many people have heard of AST and ALT. These are enzymes that are typically inside of a cell. They leak out into the blood or the serum when you have an injury to the cell. In our case, we're looking at them very much from a skeletal muscle injury, but we do know they can also come from other cells like liver cells as well. As we look at titin, for example, that's one that people may not have heard as much about. Titin fragments are actually pieces of inside of the protein, similar to that to dystrophin. It is part of the structural apparatus. And when there's injury again to the muscle membrane, they can be released into the serum and urine with that damage. LDH is also an intracellular compound. It gets released into the blood as well with tissue damage. And again, it's from many different types of cells, but we do see this also coming from our skeletal muscle. CK, I think many people have heard about, and this obviously can be and is elevated in Duchenne's, but we do want to watch that because that's a result of that muscle damage and leakage also. If we look at the heart more than the skeletal muscle, the cardiac troponin I is again, a chemical that we find inside the cardiac muscle that gets released when you have cardiac cell injury. Now, from literature, some patients who have Duchenne's have this at baseline elevations, but not all. One that is not coming into the serum, but that we actually see on staining in the muscle, similar to what we saw previously, as Pat was describing, is EMHC. And I'll actually discuss that a little bit in the future. But this is actually expressed in newly generated dystrophic muscle. And we see this on a muscle biopsy, for example. So if we go to the next slide, we can talk a little bit more about EMHC. This is actually elevated when dystrophic muscles have breakdown and regeneration. So as a muscle fiber deteriorates, muscle stem cells are activated and they replace these damaged fibers. And during this, what we call a regenerative process, newly formed muscle fibers can express this embryonic myosin heavy chain. So as you can see from the timing, we have degeneration over a couple of days, inflammation, regeneration, maturation, and then functional recovery. Now, during the regenerative process, these newly formed muscle fibers express EMHC, and high EMHC protein levels in muscle biopsies indicate active muscle regeneration. So in Duchenne's, as muscle regeneration cannot keep up with the breakdown, this leads to progressive weakness over time. But because they have this consistent turnover and changes, these muscle fibers often remain in an immature state, and so they have persistent EMHC expression due to this muscle injury and incomplete repair in Duchenne's. Because of this, we feel that upregulated EMHC in dystrophic muscles makes it a potential biomarker for disease progression and responses to therapies. If we go to the next slide, we'll see that as we look across the 90-day mark for the first three participants treated with SG003, we see a trend that is congruent across these. As mentioned earlier, CK, LDH, AST, ALT, and serum titin all showed decreases over this time period in these three participants. In addition, the histologic EMHC from the muscle biopsies also decreased, as well as that serum troponin. By monitoring these muscle integrity biomarkers, it allows us to see that trending approach for the disease trajectory and gives more indication to correlate with the muscle biopsies themselves. Next slide. So these improvements in biomarkers for muscle integrity that we saw were also seen otherwise. On the left hand, you'll see the microdystrophin expression and NOS activity that it was discussed prior, both the baseline and the day 90. What we can also see is that muscle breakdown biomarker, titin, for example, also decreased. And in this case, it was down by 42% from baseline to day 90. And in addition, as we look at that EMHC positive fibers, they also decreased from baseline to day 90 by 59%. And by monitoring these multiple biomarkers, again, we're able to show that across all of them, there's a trend in this disease trajectory that we're very excited for. Next slide. Now, beyond just the skeletal muscle, we also know that a loss of dystrophin leads to progressive heart muscle and cardiac issues. I won't go through all this data because I'm sure as the listeners here are very well aware of the disease progression of cardiomyopathy. But we do know that cardiac tissue has some regenerative capabilities and that by the age of 25, only about 1% of cardiomyocytes will turn over annually. We also know that early troponin elevation is predictive of severe cardiac diseases and neuromuscular diseases. So a troponin level of over that 7.6 is correlated with a threefold increase in cardiac risk. And thus, the question and what we're looking at is how do these troponin levels change over time? And early detection of changes in heart using troponin may inform interventions on disease progression, quality of life, and lower the risk of severe cardiomyopathy. Next slide. Given this, as we look at our AAV-SLB 101, we can see on the left-hand side, when it was tested in cell cardiomyocytes, there was an increased expression of the capsid compared to the AAV9. As we look at our first three boys in that were dosed and look at their ejection fractions, their left ventricular ejection fraction, we did notice that overall, the mean baseline cardiac function increased by 8% from a baseline of 56%. Now, participant 3 had only reached day 90, not day 180, and so we'll continue to follow this trend over time. But we're very excited and encouraged by these effects because we do know that left ventricular ejection fraction has been shown to correlate with clinical outcomes as well. So early data, but something that we're very excited to be able to share and to follow along. Next slide. Now, in addition to this, we had one participant and participant number 3 only, so it is limited to that, but we did see a reduction in the troponin by 36%, or about a third, from baseline to the day 90 as well. And as I've discussed before, the cardiac troponin I is released during this myocardial cell injury, so having a reduction is something that's exciting and a trend that we're happy to see. We know that reducing troponin concentrations by over 25% has been shown in published data to result in a 5-fold greater reduction in the risk of cardiac events. And this does also align with the positive and exciting information we have from the ejection fractions as well. Next slide. Now, moving away from some of that data into the safety data, we are looking now, rather than the 3 patients I mentioned before, but all 6 participants who had reached this time when we looked at the data. Now, in our study, we were only using prophylactic glucocorticoids, no need for intensive immunomodulation. All treatment-related adverse events resolved with no long-term effects. They began within the days following treatment, and they resolved within weeks. We've had no SAEs, serious adverse events, and no kidney injury that was observed. This is sometimes called TMA or HUS otherwise. We did have one adverse event of special interest, and this is a episode, mild, what's called a CTCA grade 1. I'll talk about the CTCAE grading in a bit. And this is a transient elevated of troponin I. Now, we purposely call out this as a special event because we wanted to see and follow these. But in this patient, there was no EKG or echo changes. There was actually no clinical evidence of heart inflammation, also called myocarditis. So it was just a lab elevation that also resolved. Now, the most common adverse events in the study were nausea and vomiting. They've all resolved. We did see patients having low platelet levels. It's called thrombocytopenia observed. One level was low enough to be called a grade 3 CTCA grading. And when I mentioned that CTCA grading, that's actually a standardized grading scale looking at adverse events across all different type of events. It's a way to actually look at differences in how significant the event was, not necessarily serious, but how significant the event was. Now, going back to our patients with thrombocytopenia, all of them, though, did return to within a normal laboratory range with no clinical symptoms. They had no signs of increased bleeding. They had no evidence of breakdown of red blood cells called hemolysis. We did not need to use eculizumab. There were no steroids or other agents used, and the patients didn't require hospitalizations or interventions as well. So while they dropped, they did also recover very well. We did have some what we call infusion-related hypersensitive reactions. These actually all resolved. One, again, had a little bit stronger symptoms with some prolonged fever, and that was why it was called that grade 3, but also resolved as well. And looking at the liver, which is very important, we had no adverse events of liver injury. Overall, as I showed before, the AST and ALT, which are both liver and skeletal muscle, had just consistent declines and no GGT increases. GGT is a little bit more specific of an enzyme in liver cells that if it increases in the blood, would indicate there's more signs of liver injury as well. So overall, the adverse events were expected and consistent with AAV gene therapy. Next slide. So where are we today? Well, here's a current summary of what we've talked about. So the initial muscle biopsy results for the first 2 patients reached day 90, and we saw on average a microdystrophin expression of 110% to 108% based off the technique that was used. The average microdystrophin percent positive fibers was 78%, and we had an average beta-glycan positive percent fibers of 70%. Overall, that average nNOS positive fibers was 42%, and the genome copies per nucleus was 18.7. As Pat mentioned before, those all correlated well across the 3 participants. There were also these biomarkers of muscle integrity that in the first 3 participants reaching day 90 showed consistent improvements across all 7 biomarkers, with no serious adverse events in the first 6 participants treated after the data cut off on that February 11th. Most of the adverse events were observed were nausea, vomiting, thrombocytopenia, but as mentioned in the previous slide, they all resolved. So we're excited with clearance of both UK and Italy clinical trial applications, and with upcoming activities adding additional trial sites by the end of this year and continuing participant screening and dosing, we're very enthusiastic and excited by this data and being able to move forward. Next slide. Here you'll see our currently recruiting trial sites. I will call your attention, given the audience here, to the last one at Sick Kids right now. And of note, there is a QR code down at the bottom. Please feel free to use that. We do try to keep that updated. It goes to clinicaltrials.gov, which will have you a consistent updated version of who is recruiting at this time. And with that, we've opened to questions.
Hi, first, thank you all very much. That was a wonderful presentation. Lots of encouraging stuff in there. So very exciting. We do have a few questions in the Q&A. So just let me open these up.
It looks like questions related to inclusion and exclusion. So maybe, Pat, if we want to go back to our slides for eligibility criteria, we can just briefly go through that again, just to make sure it's clear.
Absolutely.
Thank you.
And so maybe even without going right into the questions, if they're coming up still, as we mentioned, this is our first in human study. So we are starting a bit smaller and then hope to expand out from there. So currently the ages that we are enrolling are 4 to 11 years of age. These specific genetic variants would be the only ones that are excluded, which are deletions in exons 1 to 11 or 42 to 45. And again, just as a reminder, it's if there's any piece of these exons that are in a deletion mutation, they would be excluded. And then otherwise, prior treatments that would be excluded are just gene therapies, but anything else really would be a washout period that we're requesting.
Thank you. Annie, are you able to see the questions?
Yes, there's also a question about the difference between 003 and Elevidys.
Oh, sure. So I think one of the questions is always going to be in just getting an understanding of how each of the products either compare or differ from one another. And so we have no head-to-head data between these two products. And of course, our SGT-003 is investigational. So this is our first time that we are looking at it in these participants that we have described today. With that, some of the differences overall are going to be really those pieces that we talked about earlier on what goes into a gene therapy construct itself. And so I'll just emphasize what's in, again, the SGT-003 itself, which is this specific unique microdystrophin transgene and the use of this muscle tropic capsid that's getting better targeting to the muscle. So some of the unique aspects that are part of this therapy itself.
Also a question, Pat, in the chat about the exclusion, the current exclusion of 45 deletion. And if we have any more information about perhaps what we want to see, timing, when we think we might be able to relax that mutation, anything you can share about that particular mutation exclusion.
Yeah, I'll start it off and then maybe Matt, if you have any thoughts on it too. I think, as I mentioned, this is a specific exclusion criteria that are unique to our construct. So every microdystrophin is made up of different pieces. And so ours actually includes this region over here, the 42 to 45, because those correspond to that nNOS binding domain. So these are exons that we're not going to find in other microdystrophins necessarily. Right now, these are really excluded just out of an abundance of caution. It's something that we do not really understand whether they would or would not be immunogenic because this hasn't been tested previously. So we're beginning with these criteria, but of course want to be able to evaluate these further and really want to learn from both the experts in the field and then others as we have more data in the clinic to be able to do that.
Yeah, I think that is something that at Solid we certainly wanted to explore more, as Pat mentioned, but we want to make sure we do it in a context of understanding also the product and being safe overall for all participants going forward. So more investigations ongoing. We'll leave it at that maybe.
Oh, there might be one more. Let me just see. Yeah, I think there's one more that's just been added. Annie, if you wouldn't, can you read that one?
I can. Matt, do you see this in the chat? Do you want to take this one?
I do. So I think this is a complex discussion and a complex question about gene therapy, and really is something that I think is couple concepts that we'll want to talk about. One is the idea that as we look at an illness and Tylenol, that is a little different in that the Tylenol is sort of masking the fever and how it works. But it does come to the question of durability of gene therapy. Some of these other questions for the community that we don't have answers yet for. But SG003 certainly is an early study. in terms of the idea of could strength be affected by overexertion, those areas, that's all information that I think as we go through the study, we'll be trying to gather more data for. At this point, we'd only have that 90-day data of a few patients. It is something I would actually encourage participants and families to go back to their provider to have these in-depth discussions as they look at therapies in general. It's really important to have that discussion of all therapies that are available and all the potential pros and cons of moving into those, especially as we talk about gene therapies, because as you see, for example, in our trial, we exclude patients who received a previous gene therapy. And so these are long-acting discussions that I think are individualized that have to be had.
Thank you. I don't see any more questions in the Q&A at this time. So please feel free to add any questions that you might have. And again, as I mentioned, if anything comes to mind for you after this evening, please feel free to contact Defeat Duchenne Canada. You can send an email to info@Defeat Duchenne Canada, and we'll be sure to redirect your question to our friends at Solid Bioscience, and I'm sure that they won't mind replying, and we'll ensure that you do get a response as quickly as possible.
Absolutely. Thank you all so much for having us. We really enjoyed having the opportunity to engage with the community, and certainly happy to take any questions that people may have after this webinar. So thank you so much. And thank you, Lisa. Thank you to the team.
Thank you. So if we just will close off, I just again want to thank you so much, Annie, Matthew, and Pat. Wonderful presentation. It's so great to receive these updates. I know the community is always hungry to hear what's happening, and thank you for also your time in and responding to some of the questions that we did receive. So for those of you who may not be as connected to Defeat Duchenne yet, please join us in social media. We have lots of information to share, lots of opportunities to engage. And also, if you haven't yet signed up for our newsletter, we have an e-newsletter, has lots of great information in it as well. Please go to our website at defeatduchenne.ca and sign up for our newsletter so that you can continue to receive great updates and information. So thank you all again. Thank you to everyone who took the time to attend tonight. And thank you again to our presenters. This has been wonderful, and we hope it's just the first of many more updates to come.
Great.
Absolutely.
Thank you so much.
Thank you so much.
Thank you, and good night, everyone.